here.
Very good morning to everybody, and I really thank the organizers for giving me this opportunity to share with you about my understanding of this new situation, the COVID-19 situation, which started throughout the world. And this situation is actually telling us and also giving us so many different types of informations. That's really amazing. And every day we are getting new informations. Now, basically, my talk will be a review of the different types of information available with respect to this particular distribution of COVID-19 among the different parts of the population. And in that respect, I will try to make my presentation finish within 20 minutes and then ask for 5 minutes questions. So I'm just waiting for Professor Reggie to start the screen sharing. It's taking a little bit time, but in the meantime, I just like to tell that every alternate day, if we look at the number of prediction regarding that when it's going to have control in different parts of the world is completely getting altered. And you can see this Southeast Asia is that area of the world which is least affected. The Africa, a majority of the part of Africa is also least affected. And if we look at this particular situation in that particular area.
Dear Professor, you are controlling your presentation or our technical team controlling? The control on your hand?
No, your technical team can control, no problem.
Okay.
No problem, yeah.
Okay, this is the right slide presentation?
Yes, but this is not coming up in my screen, yeah. Somehow I could not see it.
Now we are seeing clinical features of the infection.
Yes,
Okay, we are listening to you.
Yeah, okay. And
okay, Professor.
Yes, thank you very much. And you know that when we are talking about this and this particular clinical features, now it's very explained in this slide that what are the different types of clinical features, normally what we could see at the different stages of infection. So it's a stage 1, then the stage 2, and the stage 3 is the maximum features are actually the host response. And in that particular stage, what we could see, the cytokine storm takes place, and that cytokine storm itself gives rise to a huge burden on the system itself. And when we're talking about this, this particular cytokine storm and other, which is related to the immunity. Now, at the very beginning, if you look at this initial stages of infection, we could classify the systems into the people who are responders, who are actually getting this infection, and they are symptomatic. And other than that, we have the asymptomatic patients also. And in that asymptomatic patients, if you look at again, we have some behave as a pre-symptomatic, some are not at all infected. So this whole population where there is no symptoms, they can be divided into 3 categories. And in that particular case, it is very important to find it out what we should do. Next slide, please. And if you can mention what is in the slide. Yes. Hello. Yeah, next slide, please.
Kongre odası hocamız orada sizin sesinizi duyamıyor. Mikrofonunuz kapalı. Kongre odamızda.
Evet, şimdi bir saniye hemen ilgileneceğim. Bir saniye lütfen.
If you look at this alveoli in the different stages of the infection, the free alveoli versus the one where there are started the virus have actually started to accumulate in that, and then followed by that, the whole alveoli is actually filled up with the fluid, and that actually completely disrupts the whole gas balance, and the oxygen carbon dioxide balance in the lungs is completely. Now, all these things are happening because of the alveoli. You have the ACE2 receptor maximum, and the virus actually enters into the body through this receptor ACE2. So the status of ACE2 receptor is becoming very, very important. And if you look at the status of ACE2 receptor, right now people are started to work on this, and it was found. Vivian, is there any problem? Hello? Hello? Hello, is there any problem?
No. We are following you now. Now we are seeing human coronavirus presentation slide. Is correct?
Yes.
Okay, that's good.
So this is actually the self-explanatory slide. So we can move on to the next slide. Yes, in this slide you can see.
Yes, we are seeing now.
Yes. So what is the content? You can tell us.
The life cycle of SARS-CoV.
Yes. So this is the one which I just wanted to mention to you. These are already known to everybody. I just kept it to show that this ACE2 receptor's important role to play. Now, when this particular virus gets engulfed and it goes into the endosome, and in the endosome after the fusion, because of the acidified environment, the virus loses its membrane and the RNA comes out in the cytoplasm and starts its replication. It's protein synthesis. First of all, it's protein synthesis, and those proteins are used for replication. And then the virus gets accumulated within the cell, and almost 10,000 viruses, or more than that, actually accumulates within one cell, and it comes out of the cell. And during this particular period of time, these particular cell starts are actually, if you look at the antigen-presenting cells, APC, which actually shows these viral peptides on their surface. And these viral peptides on the surface could be recognized by the neighboring cells. Then what happens? That itself tells that cell that this is an infected cell within that particular niche environment. And in that way, they can make their cells. So immunity in the system comes in that way. And then through the B cell and the T cell immunity, because the cytotoxic T cells are going to be there, as well as the B cells are there, which are actually going to the memory cells. So this is how the cytotoxic cells can actually destroy the viruses. And so that is actually takes place in the people who can fight the virus and will not allow the virus to spread within him. But the people who cannot do this, restrict the virus at this stage, they have to succumb to the viral effect, and they goes to the secondary stages of infection, to the tertiary stages of infection, depending on the viral load in the system. And when the load is very high, the virus also coming out, they can also fly and they can also move on inside our system and can start actually going into the different organs where they could find these ACE2 receptors. And there are several organs where actually you can find the ACE2 receptors are present. Next slide, please.
Professor, we are seeing now SARS-CoV and SARS-CoV-2. This is correct?
Yeah.
Or
yes, absolutely correct. Yeah.
Okay.
So this particular life cycle when we are talking about, you can find that these are almost similar what we find in case of the SARS, which actually outbreak in the year 2002. And in that particular period of time, the life cycle remains a similar type, but this receptor interactions and other things are actually different. So if we this is just to give you some comparison between the SARS-CoV and the SARS-CoV-2, why we call this as a new virus, because it has got, though many of these things are similar, but it also has got dissimilarities into this. Next slide. Next slide.
This is correct. In your presentation, you are seeing we are seeing now SARS-CoV infection can spread faster. Professor.
Yeah, this is all right. If we can move on to the next slide.
Diğerine git abi. Okay. Yok bekle. Dur bekle. Now we are checking the spike protein activation for the infection of lung cells is a graphic ACE2
Yes.
virus attachment. We should
Yes.
stop here or next slide?
No, next slide, because this is also what I mentioned to you right now regarding that in the lungs, what's happening actually.
Yeah, okay. Okay. And now we are seeing chronology of events during SARS-CoV-2 infection.
Yeah, this is also, but see, you just have a look at it because this part I have explained. And once you look at it, just move on to the next slide and tell me what is in that.
Bir daha geç.
So here I try to explain about this whole thing, whatever I actually very quickly actually gone through, and I understand that from the chats also I couldn't understand because these are in the, your language. I couldn't follow those questions and others. But anyway, I hope I could make some sense.
Yeah, exactly. Dur abi burada.
So next slide. Next slide, please.
Dear Professor, now we are seeing host cell translation changes upon SARS-CoV-2 infection. This is correct?
Exactly. So
Okay.
here I have listed that what are the different translational system of the host gets affected after the SARS infection. Now this is also going to tell us about the epigenetics, because the host, that means you understand that this is a host response to the SARS, and host response is actually affected because its translation gets completely affected, and it gets affected because of these are the different proteins and genes which are getting affected by this. Now these genes have got their own SNPs. And based on the SNPs, if you study the whole thing, it is very interesting to find out that these SNPs differences in different populations can also gives us the different response to the virus particle itself, virus infection itself. So the topic is actually the response of the host towards its infection. Some gets the infection, which is totally leading to death, and some people can actually fight it out, and some people can survive out of it. And in that particular case, earlier we have an particular things in our mind regarding the aged population versus a younger population, or the population which has got different types of diseases, and what happens in not in that particular case. All these things are coming into, if you look at this particular host translation effect. So these are the genes. I think our research can actually concentrate heavily on this to understand these differences in responses. Next slide, please.
Professor, now your screen is what are you seeing now on your screen?
Yeah, please tell me. Yeah.
You are seeing same slide, or we change the slide?
Next slide. The same slide I could see. So next slide, actually, we have to go. Over here, you can see a box appeared. So that I mentioned already. So next slide, please.
Okay. Burada dur abi, bir dakika, çok gittim. Professor, now we are seeing possible contribution of hyperactivated monocytes to coagulation in COVID-19. This is correct?
Yes. So
Okay.
these activated monocytes actually played very important role also in the responses. As I'm mentioning right now, I'm trying to get a relationship with the epigenetics part. So this activated monocytes and the response of the monocytes, how actually they are going to get activated, that also depends on the epigenetic pattern of the system. And people are studying this and getting very interesting results out of it also. Next slide, please.
Respiratory viruses and histone modifications now we are seeing.
Just now what I mentioned, that epigenetics pattern are actually getting differentiated, isn't it? Now, that is also related with the, that histone modification. And here, actually, the informations what is available regarding the histone modification, where this actually affects our epigenetics and how it is affecting our epigenetics or our own pattern, differential population, difference between the populations, difference between the different age population, difference between the disease population, you can see. Can you get my point? That this is actually the histone methylation, histone modifications, which are very important with respect to the epigenetics part. Is that clear?
Yes, exactly.
If it is clear, we can move on to the next.
Professor, now we are looking at the COVID-19 fatality risk.
Yes. So you can see that the risks are actually listed over here, and thereby what I just mentioned to you, different differential conditions and others. It was found that even in cases of certain cancer patients, how actually the number of ACE2 receptors also changes. It's not epigenetics, but the number of ACE2 receptor changes also. In different disease conditions, we could see the ACE2 receptors increase. And if there is an increase in ACE2 receptor, what happens? That the virus load in the body at the initial states can be very high. So number of cells get infected, and we can see a huge effect on before our immune system respond to that. So this is actually one of the reasons why we could see the differences. And over here, I just want to mention there is another gene called ACE1, which actually controls the level of ACE2 receptors. And it controls, that means when there is an overexpression of ACE2, ACE1 suppresses it. But in the white population, in many places in the Western world and the European world, it was found that this ACE1 expression is less. So if ACE1 expression is less, then ACE2 expression cannot be controlled. And they are going to have the same virus, same infection, but it will have a major effect on them. Next slide, please.
Professor, now we are looking at the comparative analysis of COVID-19 case fatality rates by country, sex, and age. This is correct or previous?
Yes, it's absolutely correct. This is just to give you the information which is available to us till yesterday. So if you look at based on age, based on the different countries and others, this is a comparative table, which is very important right now for our research itself, that how the epigenetics pattern of these differential age, different populations are also could be correlated. Direct correlation till not we could find, but that's a subject of the research. Please go through this slide very carefully and you can see really what is what.
Professor, now, now
we can move on to the next.
Yeah, now we are seeing known sex differences that may impact immune responses to SARS-CoV-2. This is correct slide, or we should next one? We should go to next one.
And I just want to again move on to the next slide, please.
Bir tane geç. Professor, now did you see our screen? On your screen, did you see the presentation now?
Yes, no. Can you tell me what is there?
Now we are looking at the why does COVID-19 disproportionately affect the older people?
This is the thing which I told you right now is being, I tried to put that in this particular slide.
Okay.
Remember I mentioned about these things.
Yes, yes, yes.
Just
Now we
go
are
through and this is actually the things what, yes.
okay. We are going next slide. Next.
Yes, yes.
How do you need? Now, age-related changes that increase COVID-19 susceptibility.
So this is again a continuation of the previous slide that
Okay.
why the increased susceptibility.
Yes, okay. Bir dahakine. Literature, creation of genes associated with COVID-19 morbidities.
Yeah. So this is actually the one I'm mentioning, that comorbidities. So morbidity is there and the comorbidity. That means patients are actually suffering from diabetes. Patients are suffering from heart disorders. Patients are suffering from different types of cancers. All these particular patients, these are actually, when gets infected, how they are most actually prone to infection because of the reason what I mentioned to you regarding the ACE2 receptor numbers, regarding the treatment of that, and that itself reflects when you look at this particular thing. Okay?
Yes, okay.
Next. So when we talk about this relationship with the nutrition and others, this is also actually comes with it. The nutrition also gives us a particular sense of the things. So how actually the nutrition can boost the immunity in one hand, and the other hand, it can also tell us to get out of these different types of disorders, which are actually helping the situation as a comorbid situation. Next.
Now we are looking at the mechanisms that might increase susceptibility to and severity of COVID-19 in lupus patients.
Next, because I think I have finished my time. So I think I have gone almost to the last stage, and I want to actually end my lecture because I don't want to go further, but I really like to have questions.
Conclusion.
Questions,
Professor, yeah, you have a couple of minutes before finishing. If you want, you can conclude it, or we can go to. Okay, we are listening.
Interact because the presentation is with you. You can just next few slides are actually basically ultimately going to the different bibliography and the other things where from these are being taken and other things. So main points have been covered already. So I just like to have the questions because that is most important. One question I've seen, I told already about the nutrition part, but if there is other question, because some questions I could not understand. So over here, the non-coding RNA, it's telomerizing. No, these are actually, these are, I could not understand the questions actually. So if you can go and answer the
Professor, now you are checking the questions part?
Yeah, question part. I have seen the different types of. Why don't you ask me the questions? I can answer.
Professor, we have a question and answer part end of this section. After two presenters.
The problem is actually I have to go for another meeting actually. That's the reason if I was not there. So if there is immediately certain questions comes, otherwise if I was there, I shall answer the question. But if I am not there, you can actually email me and I can send you the answers. But anyway, I like to thank all of you with our traditional namaskar. And if you can go over through my slides, I tried my best to accumulate all the informations